Hello everybody, my name is Bauer and I'm the head of the Combined Lab at the University of Surrey. I'm also the head of the NICE research group at Surrey. First of all, I would like to thank the organizers for having me and for giving the opportunity for me to talk about our research on the diagnosis of certain neurological conditions such as Alzheimer's disease. So if one is interested into the diagnosis of neurological conditions, then one has to take into account the structure and organization and function of the brain. The brain is extremely complex, as you will know. Here is a three-dimensional visualization obtained from diffusion tensor imaging, where one can see the different fiber tracts with different colors indicating different orientations. So blue would be vertical and green would be horizontal. And so what is immediately clear is that this structure, so these are not functional data, but really information on anatomical, structural data. And one can see that this is very complex. And so we use computers and machine learning methods in order to assess certain aspects of the brain. And one can do that in different ways. I mean, one approach would be a so-called model-based approach, where one has some kind of model, computational mathematical model that captures processes. And that also includes some type of knowledge, background knowledge, in order to create those models. This can be, for example, differential equations. And then another approach, which is at the other extreme, would be to use an entirely data-driven approach. So one creates a statistical model from data. And of course, there are different varieties here. These are like two extremes. But what is important in all of those approaches is that, especially from a biomedical perspective, is that they should be explainable. So the AI should be explainable, because this allows for trust and transparency in the diagnosis. It allows for accountability, especially in a healthcare setting. And also, if there are certain issues like bugs and so on, then explainability is crucial. And also from a legal perspective, including, for example, GDPR, explainable AI is very important. And so we don't want to just look into the black box, so to say, but we want to really make sense of what is inside. Now, I would like to give you an example of a model-based approach first. So in this paper here that we published a couple of years ago, we looked into Alzheimer's disease, and in particular, we looked into the structure of the brain, the connections of the brain as obtained from diffusion tensor imaging, and we used machine learning in order to differentiate, control healthy brain networks from Alzheimer's disease brain networks in order to create some model. And here, what is important or very distinguishes us from other approaches, that we created also a model of how those brain networks change over time while aging. So both in the normal aging scenario as well as in the Alzheimer's disease aging scenario, we created models of how those connections change, because it is known and very well established that during aging, those brain networks change significantly. And so what we did is we started from experimental data, and we created simulated stages, 15 stages in particular, which indicates approximately 15 years. And so we simulated how the healthy brain ages and how the Alzheimer's disease brain ages, in order to then check at what stage can we really differentiate them significantly from one another. Also, we used so-called complex network properties. If you're interested, this paper here is very informative on complex network properties and measures of brain connectivity. And so we use this as a tool to capture characteristics of the brain networks. And what we then did is we used machine learning to identify the complex network measures most important to differentiate between Alzheimer's disease and healthy brain networks. And here you can see some of those features, so closeness and strength of nodes, or the eigenvector, are all important complex network features that are very informative for such a diagnosis. And we could also pinpoint the regions which are very informative. And as you can see here, and as you may know, some of those regions are very well aligned with what previously has been already established. So that's very reassuring to see that these kind of methods allow to also reproduce some of the previously known results. And as I mentioned, we are simulating this over 15 stages, which is approximately 15 years. And what we then could show is that when we use this simulation, then the trajectories become significantly different at approximately -5 or -4 years before current clinical diagnosis. So it is known that Alzheimer's disease takes many years, usually, until it becomes clinically diagnosable. And here we could show that with our method, or our results indicated that with our method, we could do this most likely a bit earlier, several years earlier, by looking at the strength of the connections and by using those signatures that I mentioned before, those complex network features. And so this is also good to see that in agreement with intuition, that at the very early stages, of course, the p-values will be not significant, but then at the more later stages, but still early on during disease progression, we can, with this method, differentiate Alzheimer's disease networks in simulation from healthy brain networks. So this is one example of where we used explainable approaches in order to diagnose Alzheimer's disease just by looking at the brain structure. But then we may also want to go into more detailed, more higher resolution approaches of models. And so we created the BioDynamo consortium, which comprises these 8 organizations. And we published a software called BioDynamo that allows to simulate complex biological tissues, including neural tissues, over time in a very performing manner. And importantly, this software is open source, so it doesn't cost anything. Everyone can download it, can also contribute to it. And with BioDynamo, we can simulate on a cellular scale. So each of those spheres here is a cell body. We can simulate their behaviors, biological behaviors, in 3 dimensions. And this approach is called agent-based modeling because each of those cell bodies is an autonomous agent. And as you can see here, they can, for example, produce chemicals that then diffuse in space. They can obey certain rules of behavior, like clustering together and so on. And also they can see, this software can be used to simulate neural tissue growth. So here is one of the early simulations we did with BioDynamo, where we have millions of on the order of 1 million cell elements that can then interact also with one another mechanically or via chemicals. And electrical activity is also something we are currently looking into. And so this is a very powerful approach to model brain development, for instance, or can also be used for degeneration in principle. And so if you're interested in this approach or tool, then don't hesitate to have a look into the website, www.biodynamo.org, or feel free to email me. And another approach that we have used in the recent past is a more statistically driven approach. So we don't start with some kind of model, but instead we look into retinal images from a large number of individuals. So here you see such a retinal image, and in this approach, we have a lot of advantages because it's a simple technique to learn how to take retinal images from someone. So it's not a very difficult thing to achieve, and this is of course then also very cost efficient. So it's very different, for example, from MRI, where you need very expensive equipment and training in order to get MRI data from a subject. Here you can do this much more easily, and it's also very quick. Takes seconds to take such an image, and it's non-invasive. Which is very important. So there is no issues with regards to some biopsies, or you don't need this with retinal images, and so it's also not surprising that therefore there is a high patient compliance, in contrast to other methods. Like in MRI, for example, some people may be claustrophobic, which can be an issue, or they may have in Parkinson's disease can have a tremor, which can then also affect, of course, the acquisition of data. So in addition to those advantages, retinal images also, because of this many advantages, they also allow monitoring over time. So you can very easily do this several times in order to monitor any changes over time. And due to these advantages, people have in the past looked into retinal images, in particular retinal fundus images. And so here, for example, this paper here, the researchers showed that one can detect a number of diseases, including, for example, cardiovascular issues from retinal images, but these are often not really explainable. And so what we did is we looked into retinal image analysis by using some computer vision algorithms. So we segmented, for example, the vessels, we classified arteries and veins, and then we used some AI methods to diagnose Alzheimer's disease and Parkinson's disease and neurovascular dementia. So these are some of our earliest papers that we published. And the way this works is that we create around the optic disc this kind of zones, and then we can extract features dependent on the zones. So different kinds of zones lead to different kinds of features, or the features depend on the zones. And we came up with around 9 features that can be then automatically extracted from those retinal images. So this is not an AI method like in deep learning, where one feeds in an image and gets out the diagnosis. This is different. So it extracts explainable features from the retinal images in order then to create a model that is trained with the data, the labeled data. We had data from the UK Biobank, and we then also used a very innovative AI method called inductive logic programming, which is different to black box methods like many artificial neural networks. It allows to learn logic-based rules from examples and from background knowledge. So one can incorporate into these models also certain types of background knowledge. And very important, it allows them to learn structured hypotheses based on the data. So this method allows to generate hypotheses that are explainable and that then can be experimentally tested. And so ILP is really very well suited for explainable AI approaches. And so we have published recently at the IEEE conference on artificial intelligence a study where we used ILP with those features in order to really show how powerful this approach is. Here you see the results for Alzheimer's disease diagnosis, where on the x-axis we have different numbers of examples, and we of those images. And then on the y-axis, you see the accuracy. And you can clearly see here that with increasing number of examples, the performance of this method here, PyGo, which is an ILP method, achieves really very high levels and really outperforms other methods. The other best methods are also ILP methods here, but as you can see here, those methods here are really still significantly lower than PyGo, which achieves a performance in the 90s. And so it's a very accurate method. And as I mentioned, these ILP methods and usually also allow to derive some kind of hypothesis. I will not go into detail here, but what it essentially allows to do is that one can then create hypothesis on those measures and create some kind of ranges. So for example, here, if the variable b is within this range, then and if certain conditions are fulfilled, then the ILP method classifies a certain sample as having Alzheimer's. And so there are really certain types of rules that one can extract from this learned model that are explainable and that can then be used to generate a hypothesis that one can then investigate biomedically to see what may be the underlying kind of root cause of those relationships that have been discovered with ILP. And so if you want to learn more about this study, please have a look into this recently published paper. I think it was published just about 10 days ago or so. So overall, we can differentiate in the extreme two different types of models. We can differentiate mechanistic models where one has some kind of computation or mathematical model of what is going on in the system, in the dynamic system. And Alzheimer's disease is a dynamic process that plays out over time. And I've shown that with the structural changes that we modeled, we can capture important aspects of this Alzheimer's disease progression, which then can be used for diagnosis and, importantly, explainable diagnosis. And but then there are also data-driven models where one can use labeled data to inform a model and even extract hypotheses from them, which then could be experimentally tested. So both approaches permit explainable AI. So it's not like one is not explainable, but usually mechanistic models are more easily can be more easily be or readily be used for explainable methods. It's just that here we use really very innovative methods and leverage the advantages of ILP to derive explainable diagnosis. I've also shown you some examples of what one can do with the BioDynamo software, which is an open source software. Everyone is welcome to join, and it allows to really create very detailed models of neural tissue dynamics and other tissues or other complex systems, and so it also allows to investigate neurodevelopmental disorders, and we have worked in the past on using agent-based modeling to investigate certain neurodevelopmental diseases, like, for example, polymicrogyria, where the cortex is much thinner and is more folded. And so this is where the kind of biomarker approach comes in. So with BioDynamo, we can simulate neural development on a very detailed and high resolution scale in order to capture certain aspects of biomarkers for certain diseases, including neurodevelopmental diseases. And yeah, so if you're interested in learning more about these aspects, feel free to get in touch. And we also work in collaboration with colleagues here at Surrey on using computer vision algorithms to diagnose, for example, Alzheimer's disease or Parkinson's disease and neurovascular dementia. And so, yeah, I would like to thank you for your attention. And if you have any questions, feel free to reach out. And thanks